Hello everyone, we will be covering Chapter 8 titled The Cellular Basis of Reproduction and Inheritance. We will be covering cell division and reproduction, the eukaryotic cell cycle and mitosis, meiosis and crossing over, alterations of chromosome number and structure. Okay, so let's start with cell division and reproduction. Cell division plays many important roles in the lives of all organisms. Cell division is basically the cloning of a cell to create another cell. For example, we are constantly producing new skin cells, new hair cells, immune cells, bone cells, muscle mass, and it's always needed throughout our life. Not all cells divide though, so your brain cells, there's pretty much the majority of your brain cells do not divide, so basically the ones that you are born with are the ones you have for life. Neither do your heart cells, and so when somebody suffers a stroke or they have a heart attack, they can't really recover from the cells that died during those events, and that's why those diseases are basically hard to treat. But most of the cells in your body reproduce, and basically for all organisms, most of their cells reproduce as well. There are two types of cellular reproduction, and so now we're talking about organisms that need to duplicate themselves. So they either duplicate themselves asexually, meaning that one organism completely clones itself. It gives the exact same copy of DNA to its offspring, and that's called asexual reproduction. And there's another type of reproduction called sexual reproduction, in which there are DNA from parent cells that are mixed together and then given off to the offspring. And that basically creates variation in the offspring. And so the offspring, in this case, does not look like a clone of the parent. It looks like a mix of two parents. This here is an example of asexual reproduction. And starfish do this as well. They clone themselves. And you can clone plants as well. You can split them and two new plants will grow with the identical genetic information. But when it comes to more complex organisms, like humans for sure, and all mammalians, they undergo sexual reproduction, in which the offspring do not look like a clone of the parent. The only time you see clones in mammals is when there's twins. A twin is basically a genetic copy of two completely different cells that grow and divide differently in the womb, but they have the exact same DNA. That's the only time you see a clone, but for the most part, we are not clones of our parents. We have mixed genetic information from both our mom and dad, and that's sexual reproduction. Now, prokaryotes, which are basically the classes of bacteria and archaea, undergo binary fission, which means that they basically divide asexually. So that means that their offspring are going to be clones, and they basically split themselves in half. They divide in half. So as a cell replicates a single chromosome, the copy of the chromosome moves apart, the plasma membrane pinches inward, and a cell wall is made, and then it ends up being two cells. So that's called binary fission. So why is binary fission classified as asexual reproduction? It's classified as asexual reproduction because the offspring of, let's say, a prokaryote is the exact clone of the parent. So the mother and the daughter are basically clones of each other. So this is what it looks like. At the very top, you have a prokaryotic cell with one strand of DNA. That strand of DNA then replicates into two pieces. So now you have two exact copies of the DNA. Once those copies are made, the cell basically pinches itself. It creates like a new wall in between and then splits itself into two. And this is considered binary fission. And this is basically how offspring are made. Now you have two daughter cells. Now you have two complete cells with the exact same genetic information. And this is how it looks like under a very powerful microscope. Eukaryotic cells have two different ways of replicating themselves. They undergo mitosis and meiosis. First, we're going to cover mitosis, and we're going to cover which cells undergo mitosis. So the cells that undergo mitosis are all of them except your sex cells. So for males, the sperm do not undergo mitosis, and for females, their ovum, the eggs and the ovaries, do not undergo mitosis. They both undergo meiosis. But the rest of our cells, like our skin cells, our hair cells, immune cells, everything else, are clones of itself, and so they undergo mitosis. Basically, mitosis occurs when the cell is ready to clone itself. So a cell is not always in the mitotic phase. A cell is mostly under a phase called an S phase or an interphase. This is a picture of a cell with its basically chromatin condensed. So cells mostly have their DNA like in a spaghetti form where it's all kind of mixed up in the nucleus, But when it's ready to divide, then it separates nicely into chromosomes. Then it separates and organizes itself, but that's only when it's ready to divide. If it's not ready to divide, then it's like in a chromatin form, in a spaghetti form. So before a cell starts dividing, the chromosomes duplicate, producing sister chromatids, which contain the identical DNA. And then they are joined together. So the way that looks like is if you've ever seen the chromosomes, how they look like in the X form. The X form is a representation of two sister chromatids that are bound together at the centromere. Cell division basically involves the separation of those sister chromatids into two daughter cells. So here's a picture of two sister chromatids. The left arm is an exact duplicate of the right arm. When they are joined together at the centromere, you have an X-looking chromosome, okay? But the left is an exact duplicate of the right side. So the left arm is a duplicate of the right arm. And they're called sister chromatids. But in this case, the sister chromatids are stuck together. They're bound together at the centromere. And so that's what makes them look like an X. Although the left and the right are exact copies of each other. So here's another close-up. At the very top, you have a chromosome, a single chromosome. That single chromosome duplicates itself, and now you have two chromosomes that are bound together at the centromere, and those are called sister chromatids. And those sister chromatids are separated during mitosis to create two new cells, two new daughter cells, which have those chromosomes, which have copies of each other again. First, we'll go over the cell cycle, which is basically what a cell does during its lifetime. While it's alive, what is a cell doing? And so a cell basically has two phases. It's either in interphase or it's in the mitotic phase. Interphase is split into three, so all that you see in purple is interphase. So interphase has G1 phase, S phase, and G2 phase. They're just split up, but they're all one whole phase called interphase. So during G1 phase, there's cell growth. So basically, the cell is getting bigger and bigger. It's growing in size. And the reason why it's growing in size is because it's ready to split into two. So it has to be big enough to be able to give enough stuff to the daughter cell, basically. And so it grows in size and it produces proteins for DNA synthesis. S phase is basically DNA replicating itself, so that's called DNA synthesis, which is basically making another set of DNA. And that other set will be given to the daughter. And then there's G2 phase, in which two sets are completely formed. And once those two sets are formed, they can no longer be in their spaghetti phase, like in their chromatin phase. And what I mean by that is that they're no longer just scattered. Now they're going to be organized into little X's. How I showed you earlier when I showed you the two sister chromatids that look like an X. In this phase, in the G2 phase, the DNA is being ready to become coiled and organized. It needs to be organized so that when it's separated, it gets separated evenly. And then the actual separation of a cell, basically a cell dividing itself into two, is called mitosis. And so this is basically a map of the cell cycle. And if you notice, the interphase takes the biggest portion of the pie, meaning that a cell is mostly an interphase. And if you see that half of it is S phase, the DNA synthesis, that means that a cell spends most of its lifetime replicating its DNA, reproducing its DNA to give to its daughter cell. And you see that mitosis is just a little piece of the pie, meaning that mitosis happens a lot quicker. And so remember, mitosis is the mitotic phase. That's what it says right there, but that's the same thing as mitosis. That is the cell division phase. Here is a checkpoint question. A researcher treats cells with a chemical that prevents DNA synthesis from starting. This treatment would trap the cells in which part of the cell cycle? So a researcher uses a drug to keep DNA from reproducing a replicate of itself. So go back to the previous slide or the previous slides and think about which phase is the phase in which DNA literally replicates itself. And remember, this is the cycle that the cell spends most of its time. Now let's get into mitosis. So mitosis basically is cell division. And what I mean by cell division is basically that the cell is dividing itself into two. And basically its DNA is replicated and that DNA is given to the daughter cell. Before the DNA is separated, it is condensed and coiled up into tiny chromosomes, which look like little X's. Once it's coiled up and organized, it's basically like if you have a really messy room and you're getting ready to move out, you wouldn't want to just take the mess with you. You would want to organize it into little boxes so that it can fit into more space or into tinier space. And then it would be a lot easier for you to move. So that's the same thing what happens with DNA when DNA is coiled into chromosomes, into those little X's. They're coiled into that structure so that it can be easier to basically divide it. It can be easier to separate without losing pieces of DNA. And so this all occurs during mitosis. There are 4 phases of mitosis: prophase, metaphase, anaphase, and telophase. Telophase includes cytokinesis. And I do want you to memorize these and be familiar with it. Prophase is basically the first state of mitosis where the chromosomes are paired. And so basically the chromosomes look like X's in this case. They are coiled, packaged, and ready to go. Metaphase is when all of those little X's line up in a single file in the center of the cell. So now we are super organizing this whole event. So now these chromosomes aren't just scattered all over the nucleus, they are lined up in the center in a single row. In anaphase, there are mitotic spindles that come out and basically grab the legs of the X-looking chromosomes. They grab one side and the other side and basically pull them apart. And so that's anaphase, the pulling apart of the sister chromatids. And telophase is basically when the chromosomes are now on each end. They're now completely separated, and the cell can completely divide itself. It can split into 2 at this point, knowing that the daughter cell will have the exact same amount of DNA that the mother cell has. And cytokinesis basically goes along with telophase, but cytokinesis is the actual splitting of the cell. It's the actual event of the splitting of the cell. And this is basically how it looks like. In the very left, during interphase is not a part of mitosis. Interphase is that long phase in which the cell usually is in. So if you notice the DNA in interphase is like in a spaghetti format. If you look at the interphase on the very left, the bottom picture, where you see just purple that represents the DNA, it looks all stringy and it's just everywhere. It's not organized. That form of DNA is considered chromatin. It's called chromatin because it's basically in a disorganized form, which is fine. That disorganized form works perfectly for the cell when it's not undergoing mitosis. However, when it prepares for mitosis, the chromatin is no longer disorganized. During prophase, the DNA is now neatly arranged in these little X's into a sister chromatid. So the chromosomes are bound together into sister chromatid. At this point, the DNA is already replicated. That's the only reason why you have a sister chromatid, because the sister chromatids are replications of each other. So at this point in prophase, DNA has been completely replicated. Metaphase or prometaphase is basically right before metaphase, but it's all the same thing. Prometaphase and metaphase go hand in hand. Prometaphase is basically the formation of the spindle microtubules, the spindle fibers, and those microtubules work as ropes. They pull apart the chromosomes in the center. But right now in full metaphase, so this is the phase right after prometaphase, but they're all the same thing. In metaphase, you'll notice that all of the sister chromatids that are bound by centromeres and they look like X's, they're all lined up in the middle. Meta meaning middle, metaphase. So that's the middle phase. And then the mitotic spindles, so you have to look at this illustration to like know what you're looking at. The mitotic spindles are basically bound to those sister chromatids, and then they pull them apart in anaphase. So anaphase is a phase in which the sister chromatids are pulled apart and now they're back into single pieces. The sister chromatids basically are exact replicates of each other. So this is two pieces of DNA that are exactly the same. So one gets pulled apart to the left side and one gets pulled apart to the right side, or you can say like the North Pole and the South Pole. And once they're completely pulled apart through anaphase by the mitotic spindles, then you enter telophase and cytokinesis. Telophase is basically the phase in which the DNA is basically on one end of the pole, let's say the North Pole, and the other DNA, the other set of sister chromatids are on the South Pole of the cell. And so at this point, the cell is ready to basically divide itself in half by cytokinesis. So cytokinesis is literally the cleavaging, the dividing itself in half. And once that's done, then you have two complete cells. You have two whole new cells with the exact same copy of DNA. And again, remember that mitosis is very important for you to create new cells, and we're creating new cells constantly, like hair cells and skin cells, and the cells on our nails and our immune system, and for repair and whatnot, and for reproduction. I mean, we're constantly undergoing mitosis. Well, reproduction, sorry, it's a meiosis phase, but mitosis is for everything else. Here's a question for you. You view an animal cell through a microscope and observe dense, duplicated chromosomes scattered throughout the cell. Which state of mitosis are you witnessing? So at this point, if you can see the little X's, so you're looking at a cell under a microscope, and you stain the DNA, and the DNA looks like a bunch of little X's. It looks like it's coiled and condensed. That means that it's ready to divide. It wouldn't be coiled and condensed otherwise. It would be in that spaghetti form. It would be like in a big mixed form. But in this case, it's not. It's organized. So if it's organized, that means it's at the beginning of mitosis, which is prophase. And here's a picture of a cell basically undergoing cell division, and the different colored stains are to represent the different types of structures. The green colors are basically the mitotic spindles, and everything that you see in blue are basically the sister chromatid that are lined up in the middle. So if the sister chromatid is lined up in the middle in this case, what phase of mitosis is it in? And if you answered metaphase, then you're correct. Now the mitotic spindles are pulling apart the sister chromatids to each pole, to the North Pole and the South Pole. And while they're being pulled apart, this is a certain phase. And this phase is called anaphase. So if you got it, good for you. Okay, and try to guess what this last phase is, which is basically the phase in which the cell divides itself, because now a copy of DNA, basically the exact copy, is now on the North Pole and on the South Pole. So you have two complete different copies on each side. And so at this point, the cell is ready to divide itself and create two cells. So this is basically the very last phase of the cell cycle, which is telophase. And the part that is splitting in the middle is called cytokinesis. This is a video that I want you to play. You just have to press play so that you can basically witness mitosis, what it would look like live. And this is another video with the same thing. Okay, so cytokinesis is different for plants and animal cells. Cytokinesis is basically when the cell divides itself into two. But in animals, cytokinesis occurs when it forms a cleavage furrow, which basically this is formed by the mitotic spindles. Whereas in plants, since they have a cell wall is created in the center to basically split the two. And here's a picture, and I know it looks like a butt, but here's a picture of a cell basically undergoing cytokinesis. This is an animal cell, where it basically pinches itself off. And it's called basically a cleavage furrow. And once it's split into two, then you have two completely different daughter cells. Whereas in plant cells, since they have cell walls, there's a cell wall that is basically created in the middle, and then that splits up the cells into two different cells.
In animal cells, cytokinesis begins with the formation of a cleavage furrow. At the site of the furrow, a ring of microfilaments contracts, much like the pulling of drawstrings. The cell is pinched in two, creating two identical daughter cells. In plant cells, cytokinesis begins when vesicles containing cell wall material collect in the middle of the cell. The vesicles fuse, forming a large sac called the cell plate. The cell plate grows outward until its membrane fuses with the plasma membrane, separating the two daughter cells. The cell plate's contents join the parental cell wall. The result is two daughter cells, each bounded by its own continuous plasma membrane and cell wall.
All right. So the rate of cell division is affected by environmental factors. So this is basically why cells divide, like what exactly tells a cell to divide itself. And it depends on a couple of things. And this is basically what's known by researchers. There's, I'm sure, plenty of more factors, but these are things that have been well studied. So for one, cells know to replicate themselves. when a cell is missing. So cells have to be touching each other. And when they're touching each other, they're completely surrounded by other cells, they'll stop dividing. But if they're missing something, like if they're missing something to the left or to the right or next to it, then that sort of gives it a cue to basically replicate another cell. And this is not how all cells replicate, by the way. This is just the case for certain types of cells. Not all cells do this. So basically, the cells that divide until they touch one another or by touching one another is called anchorage dependence and density dependent inhibition. Other cells basically divide by a signal, and that signal is called a growth factor. And so if it receives that signal by a protein, so a certain protein will come and tell it like, hey, you need to divide, then that cell will divide. And so this is how your immune cells divide. They divide by growth factors. Here is an example of an experiment that was done to understand how cells divide based on cells touching each other. So basically, a cell was cultured on a petri dish, and they noticed that the cells would continue dividing until they were completely touching each other. And once they're all completely touching each other, then they stop dividing. If you notice in the middle picture, okay, well, let's start from the top. You have anchorage, you have the beginning of cells that are not touching each other, so they keep dividing. And then on the second picture to the top, you have a single layer of cells that are all touching each other, and they'll stop at that point. The scientists basically removed a cell, and the cells noticed that they were not all touching each other, so they reproduced more to restore that and basically remain there. So this is their cue on how and when to divide. A cancer cell, if you notice in the very bottom, and we're going to talk about cancer cells in a few slides from now, is basically cells that are disobeying. So they don't care if they're surrounded by neighboring cells. They'll still continue dividing. These cells have lost the ability to understand the cues of when to divide and when not to divide. So cancer cells basically disobey the cell cycle and they continue dividing nonstop. This is basically another experiment that was done to understand how cells divide. And to the left, you have cells that are in a culture, but they're in a culture that's basically healthy for them. They can divide if they want, but they don't. And to the right, you have cells that are in that same culture, but they are given a growth factor. And once they're given that growth factor, they divide. And so this is how scientists discovered that growth factors are something that also serves as a cue to tell cells when to divide. Cancer cells basically are cells that do not obey the cell cycle for various reasons. There's various ways in which cell division can go wrong. So that's why you can't basically completely understand cancer from one type of cancer. Let's say if you study breast cancer, that doesn't mean you'll understand how all cancers work. Cancer is sort of like a broad term of basically cells going haywire. It's like a broad term for cells that are disobeying, that are no longer abiding by the cell cycle rules. They're kind of just reproducing however they want. And again, there's many, many reasons why cells do this. It's not just one. And so that's why you can't understand all cancer diseases in the same way. They all work differently. For example, maybe some cells lost their ability to reproduce by growth factors. Well, not lost their ability, but maybe they lost their ability for the cues and they just don't care. They're going to keep reproducing. Or maybe some of their cells lost their ability to know when there are other cells surrounding them, touching them. So again, it could be various reasons why cells become cancerous. Now there are benign tumors, and benign tumors are basically tumors that stay put. These are cancerous cells that stay put, and they do not spread throughout the body. They can basically disrupt whatever is surrounding them, so they have to be removed, but they're not necessarily malignant. Malignant tumors are when basically tumors not only reproduce in one area, so let's say it starts like in the breast area. Once those cells basically divide and grow into a big mass, they're going to keep dividing, they're going to keep reproducing, and they can end up entering into the bloodstream. And once they enter into the bloodstream, they can end up in other places in the body. And if they're there, they're going to keep dividing. There's just no rules for it. And those are cancerous cells, and that phase is called metastasis or metastasis. Sorry, it's pronounced metastasis. And so metastasis is when cancerous cells have now spread throughout the body. And once a person is at that point, it's very hard to treat. At that point, it's deadly. And so you want to treat cancer before it metastasizes. The treatments for cancer, or at least the current treatments, are based. So now we're going to talk about meiosis and crossing over. And meiosis is basically the sexual reproduction that only occurs in the sex organs. And the actual cells themselves are called gametes. For females, those are the ovum, the eggs basically in the ovaries. And for males, those are sperm, which are found in the testes. There's a difference between somatic cells, which are basically the cells that undergo mitosis, and sex cells, the ones that undergo meiosis. Somatic cells basically have 46 chromosomes total, like 46 little X's, and that's because they are in 23 pairs. And those pairs are called homologous pairs, meaning that one comes from mom and one comes from dad, but they carry the same genetic information. Whereas the sex cells actually only have one set of chromosomes and not homologous pairs. They don't have two sets, they just have one set. Sex cells, the only way that they can become, or they can have two sets, is when they are fertilized. So when a sperm basically joins with an egg and they both share chromosomes with each other, now you have 23 pairs. But until then, you have 23 single chromosomes. And that's the difference between the sex cells and the somatic cells. And that's why they have to divide differently. They have to replicate themselves differently because of their nature. So here is an example of basically homologous pairs. And this is also called diploid chromosomes. So this is when chromosomes are in, there's a double amount of them, okay? So what you see in the yellow circle is basically a diploid, and it's also what you would call a sister chromatids. So the arm on the left is the same as the arm on the right. And those are sister chromatids. And that's everything that is circled in yellow. Now, the two X's that are next to each other, so the one that is circled in yellow and the X that is next to it, it's not circled, but is next to it, that's called a homologous pair. A homologous pair basically has the same genetic information. So let's say if the chromosome on the left has a gene for eye color, then the chromosome on the right will also have a gene for eye color. The one on the left, let's say, came from mom, and the one on the right came from dad. And that's why they are in pairs. It's basically in pairs because you inherited a set from your mom and a set from your dad. This is an example of basically chromosomes that are not in pairs, so they are not homologous pairs. And haploids are basically when the sister chromatids are broken apart. So in this case, you see that they're not bound by a centromere. They're broken apart, so they're all in their haploid states. So when chromosomes are not in pairs, again, they're not homologous. And this is basically how DNA is found in cells that undergo meiosis. This is how DNA is found in your sex cells, basically. They're not in pairs, they're in singles. Now let's go back to the homologous chromosomes really quick, and let's go over a few terminology. So the locus, if you notice, the locus is pointing in one spot, and a locus is basically any location on a chromosome that has a gene. So let's say the locus for green eyes on the chromosome to the left will be on the same locus on the one to the right. So let's say the locus on the one on the left is the yellow stripe, exactly where the line is pointing. So let's say that locus with the yellow stripe is eye color. Well, the other chromosome that's the homologous pair, let's say that chromosome came from dad and the other one came from mom, that one will also have a gene for eye color. But even though the location of the genes are the same, it doesn't mean that the genes themselves are the same. For example, the locus that came from, let's say, your mom, the left chromosome, could be a gene for blue eye color, and the locus on the right chromosome can be a gene for brown eye color. So the actual genes themselves, even though they both code for eye color, can have variations. The centromere in the middle, so if you notice the picture that says centromere, is what basically fuses two sister chromatids together to form that X. And it is literally fusing it so that the left arm is the exact same copy as the right arm. And they're just fused together. So the left X and the right X come from two different parents. So the left X comes from mom and the left X comes from the right. But the actual chromosomes that are held together by one centromere, so if you look at one X only, the actual chromosomes that are held together by one centromere, that's a duplicate of itself. That's the same exact, so both on the left come from mom. Alright, so let's go back to gametes and meiosis. Meiosis only occurs in gametes, which are the sex cells, eggs and sperm. And just like we talked about earlier, these are cells that are in haploid state. Well, they end up in haploid state. But as they undergo meiosis, they go through an alternation of haploid and diploid stages. So you'll have in one stage, you'll have homologous chromosomes. In another stage, you'll have only a single set of chromosomes, and that's fine. It just goes through twice as many phases, but you end up with a cell that is haploid, that has only one set. So basically, haploid gametes basically only have one set of chromosomes, and the only way that they can become diploid again, the only way that they can have two sets of chromosomes again, is if a sperm cell and an egg fuse together, and that's called fertilization. And then at that point, they're back to their duplicate chromosomes. They're back to 23 pairs. And from then on, the 23 pairs, from after fertilization, all of the cell division from then on is mitosis. So before fertilization, you have meiosis. After fertilization, you have mitosis. And so from the moment that an egg basically is fertilized, from there to like the rest of its life, it's undergoing a ton of mitosis, except for its sex cells. Okay, so let's go over meiosis. It actually has twice as many phases, but the same as mitosis. So you still have interphase, in which basically DNA is replicating itself right before it enters meiosis 1 and meiosis 2. And meiosis 1 and meiosis 2 is basically the reproduction of sex cells to produce more sex cells. Basically, you start with homologous pairs. During meiosis 1, you have homologous pairs that are split. And so now let's say the one in red, the X in red, the chromosome in red, let's say you inherited that from your mom. And so that one gets split off into one cell. And then the one that was inherited from your dad, let's say, gets split off into another cell. But it's not that easy. It's more complex. These two chromosomes actually end up mixing together before they get split off. So it wouldn't be just completely only the one from your dad or only the one from your mom. But this is just the sake for simplicity. This is just for the sake for you to understand what happens during these phases. But they actually have a mix of genetic information before they split off. And at the end of meiosis 2, you have 4 cells. So instead of ending up with just 2 cells like you did in mitosis, you end up with 4 cells that are all have haploids. They all have just excuse me, a single set of DNA. So basically, meiosis occurs in two divisions. You have meiosis 1, which starts with the pairing of homologous chromosomes. Then there's crossing over, in which genetic information is exchanged between the mom chromosome and the dad chromosome. They actually mix genetic info so that way the daughter cells can have something completely different. And that's the reason why you don't look like your brother or sister, or you're not a clone of your mom, because of crossing over. And then there's meiosis 2, which is basically a repeat of DNA splitting. But in this case, you're not splitting homologous chromosomes. You're splitting that one chromosome into its sister chromatids. And then you end up with 4 haploid cells. So let's start with meiosis 1, prophase 1, which is crossing over. Well, basically, it's the formation of homologous chromosomes into pairs. And then there's crossing over. And crossing over is when genes are shuffled around. This is literally the reason why, again, you don't look like your mom or dad, or you're not a clone of anyone. I mean, you look like your mom or dad, excuse me. You're not a clone of your mom or dad. You're not a clone of your brother or sister. You have your own, we all look alike because you share genetic info, but you have your own features, and this is because of crossing over. So crossing over is the exchange of DNA between non-sister chromatids of homologous chromosomes. It's really important that you understand the terminology homologous chromosomes and sister chromatids and non-sister chromatids, because this is all going to sound like a foreign language unless you know exactly what those terms mean. So this is basically what crossing over looks like. If you notice, the one in red is, let's say you inherited that from your mom, and the one in blue, you inherited that from your dad. In crossing over, the DNA is being exchanged between the mom and the dad chromosomes. So that way there can be a nice genetic, like a recombination, so that the offspring have different traits. And the reason why organisms do this is basically for the insurance of their survival, because organisms, we live in a changing environment, and so we want to make sure that we can adapt to those changes in the environment. So it's best to have different types of offspring that have different traits to have better chances of surviving the environment, versus a clone, where a clone would have the exact same features as its predecessors, and let's say those features are not suitable for that environment, it could kill off the whole colony.
During prophase 1 of meiosis, homologous chromosomes pair up very closely, and corresponding parts of 2 non-sister chromatids may trade places. This process of crossing over creates variation by producing chromosomes that combine the genes inherited from 2 parents. Here the process produced a total of 4 genetically different gametes. There are many ways crossing over can occur. In humans, crossover events happen an average of 2 or 3 times per chromosome pair, greatly increasing the variation among eggs and sperm. Note that crossing over produces some parental gametes with chromosomes like those of the parents, and some recombinant gametes with a mixture of genes from both sets of chromosomes.
All right, so now we've talked about prophase, we talked about the crossing over events, and now prophase is also the lining up of homologous pairs, which basically end up in metaphase, where all of those homologous pairs, you'll notice that these pairs are not in a single file like in mitosis. In mitosis, instead of having 23 pairs, you have 46 singles lined up in a whole row. And then those singles are ripped apart. But in this case, the singles are not ripped apart. The pairs, the homologous pairs, are ripped apart. So if you notice, anaphase 2 is taking one X on one side of the pole and the other X on the other side of the pole. These are the pairs that it's pulling apart. Now for meiosis 2, you have to deal with the chromosomes themselves that are in sister chromatid format. So 2 sister chromatids are bound together in a centromere. They still look like an X. In meiosis 2, that X is now ripped apart into singles, just like what you would see in mitosis. So meiosis 2 is sort of like a repeat of mitosis, where you have the X's that are basically getting ready to be pulled apart in prophase 1, so everything's lining up. Metaphase, in prophase 2. Metaphase 2 is basically all of those chromosomes, the sister chromatids, are now lined up in the middle in a single file and a single row, just like you would see in mitosis. And then in anaphase, each arm is pulled apart, is ripped apart into the north and south pole. And then that basically is divided in half, and you end up with 4 cells with haploid DNA, meaning that each one just has one arm of the X. So both mitosis and meiosis begin with diploid parent cells that have chromosomes duplicated during the previous interphase. Mitosis produces 2 genetically identical diploid somatic daughter cells, whereas meiosis produces 4 genetically unique haploid gametes. So that means that the DNA of every single cell, although they're copies, they have mixed genetics. And because of the mixed genetics, you end up with 4 unique cells. Each cell can be a completely different, it can look completely different than the mother cell that it came from. And that's the reason why you don't look like your brother or sister. I mean, you don't look exactly like them. You do have some traits. You do look like them to an extent, but you're not a clone of them. And this is the reason why, because of meiosis, mainly because of the crossing over events that happened during prophase 1. So let's compare mitosis and meiosis. To the left, you'll notice that the duplicated chromosomes remain separate, whereas to the right, where it's meiosis 1, the duplicated chromosomes are basically pairing up in homologous pairs. The mom and dad pairs are close to each other, they're next to each other, whereas in mitosis, the mom and dad pairs are not next to each other. In metaphase of mitosis, the mom and dad chromosomes are lined up in a single row. They do not care to be next to each other. Whereas in meiosis 1, the mom and dad pairs are next to each other, ready to be ripped apart. But remember, in prophase and metaphase of meiosis, well, prophase is when you have crossing over, right? And so the pairs of chromosomes that are being ripped apart during metaphase, or getting ready to be ripped apart, already have different genetic DNA. They have different genetic combinations in comparison to metaphase of mitosis, where there's no recombination events. So the DNA is identical in this case. And then in anaphase and telophase of mitosis, you have the chromosomes being ripped apart into single chromatids. So the sister chromatids that were bound by a centromere are ripped apart. And this is not the case yet in anaphase and telophase 1 of meiosis 1. Basically, in this case, the homologous pairs are ripped apart. So the mom and dad X's are ripped apart. But remember, the mom and dad X's now have different genetic information because of the crossing over events. And in meiosis 2, you end up with 4 cells, each with unique DNA. So they all look, all of the DNA is not a clone of the mother. They're all unique.
In sexually reproducing organisms, 3 processes lead to most genetic variation. Independent orientation of chromosomes in meiosis, crossing over of chromosomes in meiosis, and random fertilization. Each pair of homologous chromosomes consists of 1 chromosome inherited from the father and 1 from the mother. Here we have color-coded them blue and red. Each pair of chromosomes lines up independently of the other pairs in metaphase 1 of meiosis. Here you see one of the possible arrangements and outcomes. There are 2 different ways that each chromosome pair can line up. That means that in the organism shown here, with the diploid number of 4, independent orientation of chromosomes at metaphase 1 can produce gametes with 4 different combinations of maternal and paternal chromosomes. In a human being, with 46 chromosomes, more than 8 million combinations are possible. Now let's look at how crossing over creates even more genetic variability. During prophase 1 of meiosis, homologous chromosomes pair up very closely, and corresponding parts of 2 non-sister chromatids may trade places. This process of crossing over creates variation by producing chromosomes that combine the genes inherited from 2 parents. Here the process produced a total of 4 genetically different gametes. There are many ways crossing over can occur. In humans, crossover events happen an average of 2 or 3 times per chromosome pair, greatly increasing the variation among eggs and sperm. Note that crossing over produces some parental gametes with chromosomes like those of the parents, and some recombinant gametes with a mixture of genes from both sets of chromosomes. Independent orientation and crossing over occur simultaneously during meiosis, multiplying the number of genetic variations among gametes. Because each pair of chromosomes lines up independently, and crossovers can occur almost anywhere along each pair of chromosomes, it is possible for a human being to produce an almost infinite variety of gametes. A sperm fertilizes an egg, producing a zygote. The random nature of fertilization adds to the variation arising from meiosis. Each parent is capable of producing a huge variety of genetically different gametes. The number of possible combinations among their offspring is staggering. Theoretically, one human couple is capable of conceiving a number of genetically different offspring that is far greater than the number of humans who have ever lived.
Okay, here's a question. So if you're to examine a chromosome from one of your gametes, remember your gamete is a sex cell, is it likely to look exactly like the same chromosomes from one of your skin cells? So let's say you're comparing the chromosomes of your skin cells to the chromosomes of a gamete, of a sex cell. Will the amount of chromosomes be the same? And the answer is no. The ones from the sex cell will be singles. They will not be in pairs. They will not be in homologous pairs. Whereas the ones in mitosis will be in homologous pairs. There will be 23 pairs, duplicates. So what happens when there's alterations of chromosome number and structure? So this is basically what happens when crossing over events go haywire. So one type of crossing over event that can lead to disease basically is called non-disjunction. And this is basically the failure of breaking up homologous chromosomes. So basically, instead of one homologous chromosome going to the North Pole and the other one going to the South Pole, both can just go to the North Pole instead, or both can go to the South Pole instead. So this is how that looks like. This is non-disjunction. You notice 2 homologous pairs are just ending up at one pole, and you end up with gametes that have triple the amount of chromosomes. Whereas when this is one set, right, of gametes, and the other set just has singles. Okay? And there are different types of diseases. There are diseases in which females are only born with one X chromosome, and males are, let's say, born with a Y and 2X chromosomes. And I know I didn't go over this earlier, but just as a side, there can be another version of non-disjunction, which is during meiosis 2, in which the sister chromatids are not pulled apart. They end up going to one side or the other side, and then you can end up with gametes with, again, double the amount of DNA. So in order to visualize your chromosomes, so in order for a scientist to take a look at the amount of chromosomes you have, they would basically do something that's called a karyotype, in which white blood cells are isolated, they're stimulated to grow, and then arrested at metaphase, and then you can look at them under a microscope, and basically you can count how many chromosomes are there. So in a normal karyotype, there are 23 pairs. The very last pair, the 23rd, are the sex chromosomes. And in this case, the big chromosome is the X, and the little tiny one is the Y. And so an XY is basically a male. So males have XY chromosomes, whereas females have 2 of the big ones, 2 of the left ones, 2 X chromosomes. In trisomy 21, basically the karyotype of an individual, they have basically an extra copy of a chromosome. and this is basically an extra copy on chromosome number 21. Now, these are people that can survive this type of situation, but for the most part, when there is an extra chromosome or even a missing chromosome, let's say if it was on pair 1 or pair 2, pair 3, pair 4, most likely, because an organism cannot survive under that condition, most likely a female will have a spontaneous abortion because basically the baby cannot fully form. But when there's an extra chromosome on number 21, the baby can survive the situation. It's not super detrimental to its development. And so that's why this is the most common type of, let's say, extra chromosome disease. This is the most common type simply because the baby can survive this condition. So it's not to say that it doesn't happen with the other chromosomes. It does, but the baby, like 95% of the time, will spontaneously abort. So this is the karyotype of an individual that has trisomy, which means that they have an extra chromosome at pair 21. So they have 3 sets of chromosomes instead of 2. And normally, people with this type of karyotype display certain characteristics. So basically, these characteristics are due to the developmental stages of these individuals. There are also other situations in which a person can survive either a missing chromosome or an extra chromosome. And, excuse me, and this is basically an individual that is a male but has an extra X chromosome, or is a female and has an extra X chromosome. In the case of a female with an extra X chromosome, there's actually no symptoms. The person is normal. But in the case where a male has an extra chromosome, he might end up with certain characteristics. Some tend to be more female type characteristics, like let's say enlarged breasts for males with an extra X chromosome. And these are basically the different types of diseases in which a person can survive when they either have an extra chromosome or are missing one. And so 2 X's and a Y is basically a male individual, and that's called Klinefelter syndrome. And then there's X and 2 Y's, and the male in this case is basically normal. Then there's 3 X's, and in this case the female is normal as well. And then there's Turner syndrome, in which a female is basically born with only 1 X. And in this case, she may not reproduce or make enough hormones during puberty and may be sterile, but this can be treated with certain hormones. So in other organisms, non-disjunction can basically form polyploid organisms. Basically, these are organisms with extra sets of chromosomes. And these type of errors have basically been an important part of the evolution of a new species. And so chromosome breakage can basically be in the form of deletions, duplications, inversions, or translocations. And in some cases, like for humans, that can lead to genetic disorders. But for other organisms, or just organisms in general through evolution, this can be a positive thing. But let's talk about the different types of chromosome breakage. You have deletion, in which a whole piece of DNA, like a big chunk which has a lot of genes, is basically missing. Duplication is when you basically have a chunk or a set of DNA duplicated, and so now you have twice as many genes in one chromosome. In inversion, it's when, this all happens during the crossing over events, by the way. Inversion is basically when that DNA, when it's being transferred to the other chromatid, is basically transferred in a flipped manner. It's inverted. And Reciprocal translocation is when basically a piece of DNA that should have went to another sister chromatid instead goes to a non-homologous chromosome. It goes to a completely another different chromosome. And so these are all of the different ways in which crossing over events can go haywire. So it's not just basically whole sets of chromosomes, being duplicated. It can be just pieces and chunks of the DNA being duplicated or deleted or inverted or put in the wrong chromosome. And so, okay, so by now you should be able to compare the parent-offspring relationship in asexual and sexual reproduction, explain why cell division is essential for prokaryotic and eukaryotic life, explain how daughter prokaryotic chromosomes are separated from each during binary fission, compare the structures of prokaryotic and eukaryotic chromosomes, describe the stages of the cell cycle, list the phases of mitosis and describe the events and characteristics of each phase, compare cytokinesis in animals and plants, explain how anchorage, cell density, and chemical growth factors control cell division, explain how cancerous cells are different from healthy cells, describe the functions of mitosis, explain how chromosomes are paired, distinguish between somatic cells and gametes and between diploid cells and haploid cells, explain why sexual reproduction requires meiosis, list the phases of meiosis 1 and meiosis 2, compare mitosis and meiosis, explain how genetic variation is produced in sexually reproducing organisms, explain how and why karyotyping is performed, and describe the causes and symptoms of Down syndrome.